The overall goal of the following experiment is to reproducibly transplant stem cells into the hippocampus. First, athymic rats are subjected to stereotactic radiotherapy, simulating what might occur during clinical management of brain cancer. In the lab, human neural stem cells are grown for transplantation. 1 million cells are typically required for the treatment of a single rat. 2 days following irradiation, the stem cells are slowly injected into the hippocampus at a precise location. Ultimately, the protocol shows the migration of transplanted stem cells throughout the hippocampus.
I first had the idea for these methodologies when I started to consider the prospects of using stem cells to reverse the inhibition of neurogenesis after cranial irradiation.
Generally, individuals new to this method will struggle because growth of stem cells is not trivial and it requires daily attention and care under antibiotic-free growth conditions.
Visual demonstration of this procedure will demystify the clinical process used for image reconstruction for the precise irradiation of small, well-defined brain volumes.
Demonstrating the irradiation procedure will be Dr. Dante, a clinical professor of my department. The preparation of stem cells will be demonstrated by Miss Mary Land and Catherine Tran, technicians in my lab, and Dr. Munjal, Acharya, a postdoctoral scholar, will demonstrate the procedures required for transplantation.
We utilize image-guided radiotherapy software for accurate animal positioning prior to treatment and modulated radiotherapy for targeted irradiation with minimum radiation damage to other parts of